It's with the greatest pleasure for me to introduce Professor Trudy SCHÜPBACH from Princeton University, a world-renowned developmental biologist. All of us have a polarized body plan, right? Like our back, different from the front. And how this, I think Trudy's work through a series of genetic screening actually led to our current understanding of how body access is established. And so that's why Trudy has won numerous awards and titles, and I wouldn't go over that. I think on a personal note, Trudy is my PhD advisor. So I think she is a wonderful role model for all of us. Not only we inherit like a mutant alleles from Trudy, learn how to write papers from Trudy, but also we learn how to be a good scientific citizen, to be supportive, to be generous for your trainees, for your colleagues. So with that, let's give a warm welcome to Professor Trudy SCHÜPBACH.
Thank you, Jan. It's a great pleasure to be here and to see so many faces here in the audience. I had the pleasure yesterday of meeting some of you already on a wonderful outing that we had in a boat and in the city, and I hope in the next 2 days or so, or 3 days, I will meet some more of you. In my talk today, I would like to, my talk will be in 2 parts, basically. First, I will give you a little introduction about past accomplishments that should illustrate to you how, when we work with Drosophila, we can start understanding processes that are more universal. And then the second part of my talk will be some newer work that I'm doing in collaboration with Sas Schwartzman at Princeton, where we are using, again, Drosophila, and now very specifically looking at some human diseases and how we can learn more about those, again, using Drosophila as a model system. Much of the work in my lab has centered on this pathway, which is often called the RAS-ERK signaling pathway. In our case, we in particular worked with the EGF receptor, and I'm sure most of you are familiar with the biochemistry of this pathway, where a ligand binds to the receptor, activates, phosphorylates target proteins, which then eventually recruit and activate RAS, and finally a kinase cascade through RAF, MEK, and then ERK, the ultimate target that regulates transcription, and in that sense transmits what comes from the outside, the information coming from the outside to the cell, and activate different transcriptional programs in the cell. Now, in Drosophila alone, this EGF receptor pathway is involved in many different processes during development and also even in the adult fly, starting from axis formation up to sleep or hormone stimulation and so on. And this is true not only for this pathway, but in fact, if you think about it, in animals, we have about maybe a dozen, maybe up to maybe 20, but certainly not more, signaling pathways. And they have to accomplish so much during development and then regulating processes in the adult organism. And there is this big question, basically in biology, how can organisms with just a handful of conserved signaling pathways achieve all these different things, all these different cell fates, all the different responses? So, yeah, as I said, we have mostly concentrated on this one, and I'll tell you now how we got into it, but then also what we can learn from studying something very specific happening in flies and how we can draw larger conclusions from working with model organisms. So my lab, as Jan was saying, has always been interested in understanding how in development pattern formation occurs and how the axis of the adult fly or also of the larva or the embryo are established, how the head is different from the tail, the back from the front. And what we have learned is that, in fact, the axis, the major body axis, anterior, posterior, and dorsoventral, in Drosophila, in insects, are already established when the egg is laid. So if you look here at an egg that's being laid by the mother, you can already predict that the head of the embryo will form in this part of the egg, the tail here, and the dorsal side will be where those two structures called dorsal appendages, where they insert here on the eggshell. And in fact, what you're looking at is, as I said, the eggshell, which is made during oogenesis by the follicle cells which surround each of the oocytes or each of the egg chambers. So interestingly, then one initial observation was already that the outside of the egg, meaning the follicle cells, seem to have pattern information. They seem to know that here is the anterior end, and so there should be a micropyle made here. And here is the dorsal side, so they should make dorsal appendages here. And that corresponds to how the embryo inside will develop. And so somehow, just this observation already suggested that there might be some crosstalk between the follicle cells on the outside and the embryonic pattern that's eventually established on the inside of the oocyte. Now, to get at these questions, if we go further back in oogenesis, now when the egg is being made, we can see that already during oogenesis, these axes, anterior, posterior, and dorsoventral, are being established. Again, here's the egg. Now, if we look at an egg chamber in sort of mid-oogenesis, you can also predict the anterior end, because these big cells here with these big nuclei are called nurse cells, and they connect at the anterior end of the developing oocyte. The oocyte at this stage is still pretty small, but it will grow over time. This is the anterior end, the anterior-posterior axis, and the dorsoventral axis you can predict because the oocyte nucleus, which is this blue structure here, that's the nucleus of the oocyte, which will eventually then give rise to the embryo, is at this stage very asymmetrically located close to one side of the oocyte, and wherever it is, whichever side it is close to, that's going to be the future dorsal side of the egg and embryo. Now, in order to understand more about the processes that set up these structures and determine the axis, many years ago we did a mutagenesis screen, and we were trying to find mutations that would alter the axis of either the egg or the embryo or both. Mutagenesis is nice because if you don't know, have no idea what actually the process would be, and you make mutations that affect the process, then as you analyze the mutation, find out what the gene encodes, and use then modern molecular biology to understand more, you find out things that you wouldn't have necessarily predicted. And this is what happened here. So here is just a little sample of mutant eggs laid by mutant females that had alterations in anterior, posterior, dorsoventral patterning. These eggs here we call ventralized because they have lost some of the dorsal structures, and instead ventral structures have been expanded. Here is an egg that has the opposite phenotype. Here, the follicle cells from all around the circumference of the egg have thought that they were on the dorsal side. They didn't form a ventral structure. They all were trying to make dorsal appendages. And the embryos that develop inside these eggs, which you can't see here, show the same alteration. So we have embryos in here that only make ventral structures, no dorsal structures, and then conversely here, they only make dorsal structures and have lost the ventral structures. So these mutants seemed very interesting because they affected both the follicle cell patterning on the outside and the internal patterning of the oocyte. So then we tried to clone them and find out what they were. And the ones that were the most exciting at the moment were the mutation in this gene, which we had called torpedo because of the shape of the egg. And it turned out to encode the Drosophila homologue of the EGF receptor, which in vertebrates had been characterized as a growth factor receptor. But here clearly it's involved in patterning. And then this, the mutations in this gene, which we called gherkin, which means cucumber in German, and the egg shape reminded us of sort of these pickles, little cucumbers. This gene encoded a TGF-alpha-like signaling factor, so a ligand. And in vertebrates, TGF-alpha is a ligand for the EGF receptor. And so we had a receptor and a ligand, so somehow part of a signaling pathway involved in establishing these structures. And the very nice thing that happened is after we cloned them and made antibodies against these proteins, we found that the EGF receptor is expressed in the follicle cells. It's expressed in all follicle cells on the dorsal and on the ventral side. But the ligand that binds to the receptor and activates the receptor is very tightly localized to just this region around the oocyte nucleus. And so now we can imagine how dorsoventral patterning is relayed. We have this protein here that will activate the receptor on the dorsal side, and so making the dorsal follicle cells different from the ones on the ventral side. They start expressing a different set of genes from the one on the ventral side. And so you can see how, therefore, the oocyte coordinates with the follicle cells about how the pattern is established. Okay, subsequent work by students and postdocs in the lab showed that, in fact, gherkin, this ligand, acts as what we call a long-range signal. It's produced, this is like a cross-section now through the oocyte at the anterior end. So the gherkin would be localized, the signaling factor, around the oocyte nucleus. It would activate the receptor in the overlying follicle cells, but in fact it seems to be able to diffuse to some extent and has effects on certain genes, not only on the dorsal side, but also further down. So one gene, KECON, seems not to be, that promoter seems not to be very sensitive to the amount of EGF receptor activity, so it is only then expressed in response to signaling to it on the dorsal side to a certain degree, whereas a gene like PIPE, which is in fact repressed. Here's a picture of the actual, what that actually looks like. PIPE, which is ultimately repressed by EGF receptor activities, is much more sensitive and gets repressed to a further down on the circumference, telling us therefore that different target genes seem to have different sensitivities to the level of signaling. There are more target genes that I'm not showing you here, and each of them has its own domain of expression and reactivity. we then studied this whole thing further and found that in fact the same signaling pathway, the same ligand, gherkin, and the same receptor, EGF receptor, act already earlier in oogenesis, and they early in oogenesis, in fact, the same signaling pathway sets up the anterior-posterior axis. So we have here at an early stage, when the oocyte is still very small, you can see the oocyte nucleus is right on the axis of symmetry, sitting here in the middle of the oocyte, and gherkin is expressed in, it's not localized asymmetrically yet, and at this stage, it signals to these posterior follicle cells and makes them posterior. These posterior follicle cells then send the signal back to the oocyte, which influences the oocyte cytoskeleton. It changes the polarity of the microtubules in the oocyte, and that drives the oocyte nucleus away from the posterior end, away from the middle of the oocyte, and over. It ends up then over on one side of the oocyte. Wherever it lands, the gherkin RNA and then the gherkin protein follow the nucleus, and they now, gherkin again signals now to these lateral follicle cells and instructs them to be dorsal. So then here now is, here is just a blow up to show you better what that looks like. So early, you can see how the gherkin is taken up by the, or endocytosed by the follicle cells. After it binds to the receptor on the outside, it gets endocytosed. That's how these receptor and ligands act. And you can see that here you have a pretty symmetrical distribution around the posterior end, and these follicle cells will be posterior, whereas here the nucleus is now on the side, and you can now see that it's the lateral follicle cells that mainly take up or are activated by the ligand, whereas the ventral side now is not activated anymore. So then we can ask now again, how can the same ligand receptor pair result in two different cell fates in the follicle cells? What's different between these stages? Now, in order to understand that, we did again screens, and these screens in which actually Professor Jan was involved in, and she was a graduate student. Here we were looking for mutations in the follicle cells that when the follicle cells were mutant would affect the anterior-posterior patterning. These screens were done in mosaic egg chambers. So the original screens involved mutagenizing males and then breeding them and getting females that were homozygous for a mutagenized chromosome, and then we just looked at the eggs which were laid by these females to find the mutants. That was a good screen. Obviously, it gave us the entry into the pathway and understanding, but that screen has the disadvantage that if a gene is lethal, is an essential gene that is used during development for an essential process, then you can't get homozygous females in the first screen. So you won't find that gene because you don't have an ovary to look at. So then it's better to work with this mosaic scheme where the female itself is heterozygous, and then you induce homozygous clones. And here the wild-type chromosome was marked with GFP, and when the wild-type chromosome was lost from the cell, then GFP was lost. So here, for instance, you can see there is a clone of mutant follicle cells here which don't express GFP, or here at the posterior end. And so we were looking for situations where here in the wild type at this stage, the oocyte nucleus would have migrated to the side, and gherkin protein with the nucleus would be over on the side. And here we were looking for mutants, among other things, that if the follicle cells at the posterior were mutants, the oocyte nucleus didn't migrate over to the side, but just stayed at the posterior because the microtubules weren't instructed to change their polarity. So now, again, mutagenesis is exciting because you never know what you get. And in this particular case, it was particularly surprising, and I don't have time to go into all the detail, but what Jan found is that some of these mutations that we were looking at were in fact mutations in a large machine in the cell, a pump, a proton pump called VATPase that is involved in endocytosis. And so then that was very curious at the beginning, why would endocytosis machine, a complex that has to acidify vesicles during endocytosis, why would it affect anterior-posterior patterning? But what Jan then was able to show is that these mutations really, at this stage, the major effect is that they block notch signaling. And here are some target genes that are, so notch is another signaling pathway. And at this stage of development, it should become active. And for instance, let's take the middle gene here, just as an example. So hindsight is a gene that in early oogenesis does not express hindsight, but when notch signaling is active, hindsight is turned on. But you can see in the mutant cells, those are the ones that don't express GFP, hindsight fails to be expressed. Whereas in the wild type, you can see it here marked in red. So this pump, when it's mutant and can't acidify the vesicles, it blocks notch signaling. So somehow, notch signaling requires this acidification of vesicles. Somehow, the gamma secretase, which activates notch and cleaves the intracellular domain of notch, is dependent on somehow whatever the acidification does to the protein. Maybe it changes its conformation slightly so that the secretase can cut it, or we don't quite know what structurally happens. But in any case, this was very surprising, but also very interesting. With respect to signaling, what he told us, therefore, is that the difference between early and late is that early in oogenesis, it's sort of in mid-oogenesis, we have the posterior follicle cells see two signals at the same time, EGF receptor and notch. And when both signals are active, they become posterior follicle cells. So they turn on a certain set of genes that requires the simultaneous activity of two pathways. The slightly later, notch signaling is not on anymore, and now it's only EGF receptor signaling, and that results in turning on genes like KECON or repressing pipe. So from that, then we can draw some conclusions from these, as I said, so more general conclusions about signaling. We can get different cell fates from one signaling pathway by combination of different signaling pathways. So that makes a lot of sense, right? If you have 20 different, let's say 20 different pathways, you can now combine which ones you turn on or off and get many more combinations and therefore many more possibilities of making different cell types. Another thing that helps is that you can get different cell fates by having different levels of activity. So if you have high activity of the pathway, you turn on one set of genes, and low activity, another set of genes. I don't have time to go into the experiments that led to the third conclusion, namely that the duration of activation can also induce different cell fates. These are more recent experiments that I was happy to, in fact, collaborate a little bit with Heath Johnson in Jared Tutcher's lab at the time, who had constructed an optogenetic system where they could, with blue light, induce or activate the RAS-ERK pathway. And what we saw there, that in the early embryo, when there was just a pulse of blue light in the lateral side, the gene IND was activated. But if we had a long activation with blue light, then a different gene, tailless, started to come up, and tailless is a repressor, and it would then repress IND, so that depending on how long the blue light was on, you would see different cell fates. So those are sort of more general ideas that come from the specific Drosophila, but which are also applicable to many other situations, other animals that have to develop and regulate transcription. So, as I said, in more recent years, learning from this pathway and starting to think about it, my colleague Stas Schwartzman in Princeton and in discussions, we felt since we know this pathway so well and we can test it with we have different means of testing it in flies. Flies are very amenable to genetic manipulation. We should use it to maybe study some things that are very relevant to human medicine. And so we decided to look at a class of syndromes, human patients, that are called RASopathies. They are germline mutations in the RAS-ERK pathway. So that means it affects children that when they are born with certain mutations in this pathway show a variety of phenotypes. Their facial bone structure is abnormal. They often have heart defects, skeletal abnormalities. They also have neurocognitive delays. And this is not so seldom. About 1 in 1,000 births, a child is born and has one of these phenotypes, has this phenotype. And so the clinicians at children's hospitals, like in Philadelphia, we've talked to them, to some extent, see these relatively frequently. So what are those mutations and how can flies help study them? Well, the reason maybe why they are so frequent is that there is this pathway that I told you. We have the receptor, we have these genes that are phosphorylated by the receptor, and some are positive, some are negative factors in defining the signal. And then the signal transduction also has more genes, and in practically all of them, you can have gain-of-function mutations that then lead to this overactive pathway at the end, and you end up with these RASopathies. We have concentrated on the gene MEK in flies. There's only one in flies. This is the human pathway. And it is because it sort of takes the input from all these different feedback loops and transmits the ultimate signal, how big or how small the signal is, to this factor ERK, which then goes into the nucleus and mostly affects transcription in the nucleus. It also has some other effectors in the cytoplasm. So these are, as I said, mutations, but there Many of them are gain-of-function mutations. In particular, the ones that we are looking at in MEC are gain-of-function mutations that make the pathway overly active. So we decided to look at some of them. So here, this tells you what these mutations were. The 44th amino acids, instead of here, E is changed to a G. And what Yogesh Goyal did when he came to the lab, and he was sort of co-advised by Stas and myself, is first of all really testing whether those mutations were really gain-of-function mutations, and if so, how much of a gain-of-function mutation are they? So he expressed the mutant protein or the wild-type protein, isolated it, also isolated ERK, wild-type ERK, added ATP in the test tube, and then checked for doubly phosphorylated ERK with a specific antibody that just recognizes this form of ERK, which is the activated form. So ERK gets doubly phosphorylated and then it's active. And so you can see that in wild type, the wild type control, after 90 minutes, you can just barely see a tiny little bit of double phosphorylation. However, if you now look at certain mutants that were introduced or that were made according to mutants that are known from human patients, many of them will activate quite strongly. And if Yogesh quantified it, you can see that in wild type, after here is 30 minutes, not much has happened with DP-ERK, but in, say, this mutation here, already after 5–10 minutes, you have a substantial amount of activated ERK, and the wild type amount goes down dramatically. So indeed, these mutations are gain-of-function mutations. They're ligand independent because we don't need the ligand here, or we didn't put in the ligand, and yet they can activate. They have a faster response to ligands, we can show that, and they are independent also from adapters. They don't need adapters. You can just put them naked into the test tube and they will activate. So now what do they do? That's now the question, okay, we can measure, but what effect does that have in an organism, in now the fly? And the advantage of the fly is, of course, we can engineer mutations in there and look at many flies with the same mutation and again, quantify effects. So here is just to show you where RAS-ERK pathway is active in the Drosophila embryo, and we'll initially now concentrate on that very early stage, the first activation of this pathway, which happens about 2 hours, starts 2 hours after the egg has been laid in the embryo, and there is a receptor tyrosine kinase called torso, which gets activated at the anterior and posterior end of the embryo by a factor torso-like that's localized there. And so then we have ERK activity at the 2 poles. So what Yogesh did initially is to so here you just see the pathways. We have trunk, which is the ligand, and is active at the poles. The receptor goes to ERK, and that then is responsible for the patterning of the embryo at the anterior and posterior ends. So you can see it probably best if you're not familiar with fly embryos at the posterior end, where embryos have spiracles and some other structure, and here in the mutant embryo, the whole posterior end is missing. So here again is what's known from human patients. All of these are mutations that have been sequenced in human patients, and you can see some of them have been seen in multiple patients independently. So those were the ones we've concentrated on first, and Yogesh made transgenes with these mutations and transformed them into flies, and then used what we call a UAS Gal4 system, which was active during oogenesis and made those mutant proteins so that they were present in the egg when the embryo started to develop. So then we can again look at diphosphorylated ERK. And the first thing we did is look in the middle, where normally there is no ERK activity at this stage. And when Yogesh measured, he saw that indeed, whereas in wild type, this is, we can call this 1, or that's our background measurement, but you can see there is a significant increase in activity in the mutant when he expressed this mutant protein. That is like what you basically expect from a gain of function mutation, that you get more activity. The surprise was when he measured at the ends of the embryo. This is where we normally see the signal at this stage, but when he measured, he found that in the mutant, with the mutant, the gain of function mutant, there was actually less activity than in the wild type. So whereas in the wild type, and this shows you the whole profile over the whole embryo, whereas in the wild type, you get these strong peaks at the ends. In the mutants, this is attenuated. Again, here in the middle, you see the gain of function. There is more activity, but at the ends, there's actually less activity. So it's a puzzle. What does that mean? The one thing we came up with is, first we checked whether this was just one mutation, but no, we saw this in all the mutations we looked at. Always the mutant had lower activity than the wild type at the end. It did have gain of function in the middle, but not at the end. So the one explanation we came up with was to think that there must be some type of maybe a feedback mechanism that's already operating in these embryos. Most signaling pathways have feedback, negative feedbacks, because it makes sense that the ligand or the signal comes, the receptor gets active, and then the cell wants to get ready, eventually turn down the signaling activity, get maybe rid even of the ligand or the receptor, so that it's ready to get another signal if another signal comes. And so we were thinking that one thing that we did here is, we overexpressed the mutant with the because the Gal4 system that we use in flies to express these transgenes is very, very powerful. And so we have probably 20, 50 times as much of this mutant protein than wild type. And it may, and since we expressed it already during oogenesis, it may already in oogenesis have been triggering feedback loops that were then already operating at the time when in the embryo, 2 hours later, the real signal came. And then the real signal couldn't overcome the feedback anymore because it was, was already active. And indeed, yeah, this is just sort of a schematic idea. It is known, of course, in the RAS-ERK pathway that there are some feedback loops, so we were wondering what it could be. Could it be phosphatases or internal feedback, or could it be something else that prevented this from happening at the posterior and anterior end? And some other now a new graduate student, you and a postdoc, Rob, did some transcriptome analysis, but also looked for candidate genes that might be involved, and indeed were able to find that a particular phosphatase, MKP3, seems to be mainly responsible for this attenuation at the 2 ends. So here again is, if we just express a mutant form of MEC, and you can see again the lower signaling profile at the anterior and posterior end, higher in the middle. And now if we use an RNAi construct against this phosphatase, you can see now in red, this is gone. This difference between the mutants and the wild type at the anterior and posterior end is basically gone. You also see an effect even in the middle, that now the difference is bigger than here. But of course, the most prevalent difference is here, say at the anterior end and the posterior end. And this is just a control to show that if in a wild type background you use that RNAi, it has no effect. So it's really there to block this extra signaling that we are introducing with our transgene. And this is actually a lesson now for the clinicians. And because, of course, they would like to help the patients. And so if you have a gain-of-function mutation in a gene like MEC, you'd like to give maybe the patient an inhibitor. And there are inhibitors. They're toxic, so it's not so easy. But that's sort of the idea is, okay, let's inhibit the pathway to some extent to get it back to wild type. But if you have, by chance, in a patient, a situation like this, where you have a tissue or a cell type where you turn on the signaling now too early, you might actually block the natural signal. If that natural signal comes later, it might not be able to go to the normal level, and then you actually are dealing with a loss-of-function mutation. And if you here give an inhibitor, you'll just depress it further, and that's not really a good idea. So just to tell them that, be careful. Tissues are complicated, and you really need to understand what happens in the different cell types before trying some medication that you're not quite sure what it's actually going to do. However, for us, it taught us the lesson that we have to be careful with these transgenes, that they can have sort of unexpected effects that we have to deal with. And so then we decided to do the same thing again, but this time use CRISPR and go into the endogenous gene so that we would have a normal expression level and at the normal time. And that's what, sorry, let me just kind of say I've gone through with these different conclusions, and so we'll add one more general conclusion here, that in some cell types there are feedback mechanisms to be expected that will modulate the levels and durations of the signal. Okay, so now we made these CRISPR alleles, and they had milder phenotypes. In this particular case, because now we didn't overexpress and we did not express it already early in development, you can see now the mutant is higher than the wild type at all positions. The difference with this particular allele is, as you can see, and this is true for the other alleles too, it's not that big. It's significant, but it's not such an enormous difference. It's a small but statistically significant increase, and as I said, it was true for basically all the mutants we tested quite a number of them. Now, when Rob and you looked at the effects, the first effect that they saw is that embryos often died as embryos, but not all of them. So about half of the embryos died. The other half, however, hatched out. With some delay, but nevertheless, they were able to hatch out as larvae and survived. At the next stage of development, where they were easy to count, is asking how many can pupate. Again, it's about half that, a little bit more than half, that are okay, but there is a loss of individuals that were lost as larvae. And then some of the individuals survived to adulthood, and they were sort of fine, but they had abnormalities. They had some extra wing veins. You can see here in the wild type, the wing vein is always just straight across the posterior wing vein. And here there's this extra little piece of vein. It's not very dramatic, but it's very easy to count and very easy to see. The eyes are rough. And so what we had to conclude here is that there is a great variability in phenotypes, something as geneticists that we call incomplete penetrance, and which we're, as geneticists, quite familiar with. We often have alleles with a sort of incomplete penetrance, and it's a little annoying when we have that. But this was really, indeed, in this group of mutants, there was a great variability. Individuals were lost throughout development, some as embryos, some as larvae, some as pupae, but some individuals were totally fine. They hatched as adults and were fertile. The variability of the phenotypes is also observed in humans, patients. As I said, there is a variety of characteristic things that can go wrong, either with the face or with the heart or the skeleton, but there's large variabilities and confounding to the clinicians again, because it's hard to work. And this is true for many human diseases too, that they're not fully penetrant and variable. Well, we would postulate that the surviving human patients that you see in the clinic are actually those escapers that make it to adulthood. We would think that some of these carriers die and you never know about them because they die as early embryos. On the other hand, we would also think that it is possible that some of the carriers go undetected because we have some flies that are totally fine. And we were able to confirm this working with somebody who studies this, what's called the UK Biobank exome sequences, where in the United Kingdom, they just have thousands of people, regular people, who come to the clinic in their national health system and accept that their DNA is being sequenced. It's all anonymous. And if one just looks in that database, one finds the same mutations. So there are some people there in the UK that are carriers of the same mutation that can have severe defects in other patients, but they never know. They feel fine, and maybe they have some small abnormalities, but nothing that made them go to see a clinician. So now we're starting to think about this variability. What controls then the appearance of the different phenotypes? Why are these differences there? And there are three things that one usually talks about when one sees variability. One is that there might be modifier genes in the background, or the genetic background, people would say, are different. And in humans, of course, as we all know, there is quite variability in our genetic background. So that could be the case in humans. Not so much maybe in our flies, because we made these mutations, and the way you make a CRISPR mutation is that you end up with one fly that has hopefully the right change, and then from that fly you make a line. So there's not that much variability in that particular line, but still there could be a little bit of genetic variability. Environmental factors, yes, certainly. I mean, it could be in humans as in flies, if the diet of the mother or stress of the mother or all kinds of things could influence the genotype. And then finally, what people many biologists don't like so much, could there be stochasticity, meaning just a random event? as I said, in biology, we feel like, no, there shouldn't be random events. We understand everything. This shouldn't happen. But in fact, if you think about transcription, translation, or transcription in particular, there is stochasticity there. People who look now, as it's possible now, look at transcription of a particular gene in a particular nucleus will see that sometimes there's a burst of transcription and then the nucleus shuts off again, but the neighboring nucleus may be transcribing. And so there is quite some variability there. Biochemical processes basically are based, when we calculate them, we base it on random coming together of molecules. And so there is definitely the possibility of having some random events in biology. So how do we distinguish those three problems? The stochasticity has also been discussed in humans, although again, people are not so much open to this. And we found, for instance, this paper that was published about monozygotic twins and with a mutation in neurofibromatosis, which NF1, which is a negative regulator of the pathway. So when you make a mutation, again, the pathway becomes too overly active, and they showed different phenotypes. Only one of them had scoliosis and enlarged heart valves. The other one had strabismus, hypertonia, and dental abnormalities. And here, of course, in monozygotic twins, we're thinking about, well, they have the same genotype by definition. But then the human clinicians were thinking, well, but they could, even twins that develop within the same mother can have, depending on how they are attached to the uterus, there are sometimes differences in how much nutrition one gets versus the other and so on. So they couldn't quite bring themselves to talk too much about random events, but still this was kind of an interesting case. Now, in Drosophila, we thought we can eliminate genetic variation and environmental variation by looking within one individual at right and left. So if it's nutrition, then that individual has either eaten enough or not eaten enough. So it should actually show maybe an abnormality on both sides more often than only on one. Where we looked first is in the tracheal cells, and I have to show you this. So in insects, oxygen comes to the tissues through these trachea, these big tracheal trunks, air pipes, and then they, in each segment we have these branches that, a pair of branches that innervate, or not innervate, but that oxygenate then the tissue, the underlying tissue, through these fine tubes. And in development, it's known how they are formed by the work of many labs. The pathway is known. It involves, again, the MEK-ERTH pathway. And it, in each segment of the larva, you have a right and a left branch that then send out these tubes to the tissue. When we express these MEK gain-of-function mutations in these individuals, we get cases where in the segments 3 terminal cells are formed, sometimes 4, sometimes 5. We can count them, so we can go through and look at right and left and compare what's happening. And now I'm almost done, but I need you to do a little bit of math to follow me. So what we did now is to count the frequency of these extra cells, right versus left. If we have, F is just a general frequency of extra cells, which with this particular mutation, for instance, was about 15%. So now we were testing whether there was a difference to a random distribution. So the expected distribution from random would be that you, for instance, if this is for a single side to have an extra 15%, then to have these ectopic cells on both sides on the random condition would be this F squared. Normal would be 1 minus F squared, and then on the left is F times 1 minus F and converse on the right. And you can now calculate this. For instance, F squared, 15 squared is about 225, right? So 2.3%. So then we counted, and you can see there is an enormously good correlation, between the expected under a random distribution and then the actual observed frequencies. Here's another mutation doing the same thing. It has a slightly different frequency. But again, the comparison between expected and observed is basically, if you do a chi-square, you find that there's no statistical difference between the observed. So it's totally consistent with a random distribution. We also did it for the wing, and the same thing came out, that between the expected random frequencies and the observed frequencies, there is an excellent correlation, making us therefore conclude that it is random whether or not you make an extra wing vein or not. The overall average is 13%, meaning you have a 13% chance of making an extra wing vein, but then who makes the extra wing vein is random. Sometimes on the left, sometimes on the right, and in some cases on both sides, and oftentimes not at all. Okay, so then now we'll just think about the theoretical implications. These are not graphs with real numbers, just a thinking to explain how we think about it. So the first thing is to think that we have only a small increase in the signaling output here in all of the mutations that we've looked at. So if in the wild type the signal has a distribution, this is known. Signal, if I measure signals, they're not precise. There is a distribution, often quite large distribution. But then the cell uses internally, through these feedback mechanisms, what we would call sensitivity, that ultimately results in a somewhat sharper signaling input. But still, it will be an average and then a sort of a random distribution where the cell is at that particular point and how when the signal comes and how it reacts. And in a wild type situation, that average is far away from the threshold of where you get the mutant phenotype, so that usually everybody is fine, whether they're here or here or here in the distribution. And maybe a couple of individuals will show a phenotype or not survive or not hatch out of the egg. Now, in the gain-of-function mutation, we would say there is a shift, not in the signal, but we have made a shift in sort of the sensitivity. It's more sensitive. And so the ultimate now is that we have shifted the distribution to a regime where there are more individuals going to be across the threshold for the mutant phenotype. But it's random. You could be here, again, you could be here, or you could, with a certain frequency, be here and show a phenotype. And now if you therefore think that these receptor mutations have mild gain-of-function alleles, most of the individuals at each time in development when a decision has to be made are okay, and a couple are not okay. But now we have, this will lead to what one would call the complex syndrome, or because if the pathway is used in multiple developmental events, each time you have a chance of being normal or abnormal. And if you now sum that up over time, so some of the individuals will have this extra vein, some will have these rough eyes, some will have extra trachea, some will die as embryos when the pathway is activated the first time. And so in the end, and in humans, you end up with all these phenotypes because depending at each stage, a tissue a cell may be dividing or just coming out of division and another one does not, or one transcribes a bit more or not, and depending on that, you have a phenotype or not. This is not to say that in humans, genetic background or environment has no influence. Of course, in humans, we have that. But if we take those two big influences away, which we can do in the fly, not in humans, then we see that there is also an underlying stochasticity that we can reveal in a model organism, which, as I said, can help also the clinician think about what's going on. Okay, so there are all these conclusions that I already talked about, how we get these different signaling events. And so the last one would be that in mild mutations, stochastic fluctuations can lead to variable phenotypes. And we should emphasize mild because, of course, if we have a strong, what we call a strong mutation, fully penetrant, then this whole curve would be just, because they're strong, they shift the whole curve into the where everything is mutant and every individual shows a mutant phenotype. But if we have these mild mutations, stochastic fluctuations can lead to the variable phenotypes. Okay, so now I just want to thank some of the people who were involved in the work. So the second part was done in collaboration with my colleague, Stas Schwartzman, who's fearless leader of the whole PAC group. And here is Rob, who made the CRISPR mutation. Yogesh was the one who initially made the gain-of-function mutation, expressed them with the GAL4 system. These are students, they are have been working on that tracheal system. And then I also, for the first part of my talk, of course, I have to thank my lab over the years. Here you see many of them coming together at a lab reunion. You can recognize this person here, I think. And yes, so I have to thank all of them too. And I thank you for your attention.
That's a very impressive talk. So questions from the, okay. Renjun.
Hi, Professor. Thank you so much for the fascinating seminar. My question is like a chicken egg, which appears first. Just like the gherkin protein and the nucleus move towards one side, like the dorsal. Is there any upstream signaling determining their movement, or is randomly moved to one side and determine the dorsal?
That's a funny question, because yes, we think it is random. We think that so the microtubules seem to be pushing the nucleus away, but the nucleus has an affinity for microtubules, and the microtubules are more concentrated on the side of the oocyte. And so it seems that where the microtubules come and they push this nucleus, and if it goes to one side, then that's where it will stick. The reason for thinking it's a random event there as well is that a former postdoc, Siegfried Roth, they looked at some mutants. There are mutants in myosin, and so cell division is not always complete. And so there are cases there where there are two nuclei end up in the oocyte. And if he measures where they are with respect to one another, it's random. So they're equally often 180 degrees away from one another, as 90 as close together, which would tell us, I think, that there's not other signal coming that says, okay, here is dorsal, that they can do. And then gherkin follows both nuclei, wherever they are, and then you get dorsal appendages, extra dorsal appendages, yes?
Thank you.
Yeah, anybody else?
As a student, I was always confounded by the term genetic interactions, like one gene interact with the other, and sometimes you can make two mutations to make it the phenotype fully penetrant.
Yes.
In your case, how do you explain that? If the other mutation somehow shift the spectrum?
Yes,
all
I would kind of argue that the first mutation shifts it a little bit, and then another one shifts it more, and now all of a sudden you get a more penetrant.
More penetrant. Then how can you tell it's additive or like?
I think that depends very much on the pathway, right? Whether it's going to be additive and what those genes actually do, or whether it's more a sort of a biochemical pathway where we think in terms of Michaelis-Menten curves or whatever. I think that would depend on how the interaction actually occurs.
I see. Yeah, cool.
Yes.
Thank you.
Yes, good question.
Student? Oh, okay. Tim.
Thank you very much for the wonderful talk. So I have two questions. One is about the phosphatase, in your second part, phosphatase MKP3. If I misunderstood, please correct me.
Yeah.
So it has functions when you overexpress this mutant form of MEK, but they don't do anything without this overexpression. So why there's no negative feedback at the poles of this APX?
Yes, I think that it's a good question. I mean, I don't think you can necessarily predict. Maybe I would also have predicted that if you just make RNAi against this, you would see a difference. But I think in this particular case, because this signal comes at this time, and then we measure actually pretty quickly after the signal has come. So I think if we looked at the curve, at the decay of the signaling curve, because it does decay over time, it would probably look a little different, maybe, if the phosphatase were there or not. Would maybe the biphosphorylated ERK would probably go, I would think, maybe faster away when you have the phosphatase than when you don't have the phosphatase. But in a wild type, and at the time when we measure, it hasn't had that effect yet, I think. But I agree with you. I think if we followed over a longer period of time, we should also, even in the wild type background, see a difference whether the phosphatase is there or not. And in some other tissues, it might actually make a bigger difference, depending on, yeah.
Thank you. And my second question is about your third part. You kept saying that mild phenotype of this CRISPR mutant. But you already showed that like 50% lethality in the embryo and additional 50 in larvae. It's a, in my view, it's a severe phenotype.
That's right.
And if you calculate, so I'm not good at math, but if you do this math only using escapers, then you'll lose all the severe phenotypes during this lethality.
Yes. No, right. I totally agree with you. I mean, in general, I think everybody in the room would agree that just saying lethality is not a good phenotype. In fact, it's easy to count, and that's what we did. But you'd like to actually know more. I showed you very briefly how many steps in embryogenesis this pathway is active. And so probably some of them, again, died right at the beginning. Some of them died because they didn't set up a tracheal system. Some of them died because they didn't specify the nerve cord. Some of them, you know, and we just said, okay, they're dead and we're not, but yes, I think, but I think that again, there is probably a certain probability that you don't set up the nerves or the muscles involved in both correctly, you die. You don't set up certain probability, you don't set up the tracheal system, you die. So I think when we said 50% lethality doesn't mean that there's only one process in embryogenesis and it has a 50% penetrance. I think there are multiple, and each of them has maybe 10, 15% penetrance, but then it all adds up to 50 at the end of embryogenesis. Yeah.
Thank you.
More questions from students? No? If not, let's thank Trudy for this wonderful seminar, and we have a reception outside. Okay.
Okay. Thank you.